Okay, welcome. So I'm changing gears a little bit from the epidemiology and LARC2 to stem cells, and I want to present today our stem cell program, Parkinson's disease in a Petri dish. Before I start with my presentation, I want to announce that we just received a $6.7 million grant from the California Institute for Regenerative Medicine last week. So this grant is to recruit a new stem cell scientist. It's called the CERM Leadership Award. And the person who's coming to our institute is Dr. Dennis Steingler from the University of Florida. So he'll be joining our team early next year. So that's really exciting. And he'll bring his whole lab and people, and we'll do even more great science here. So I want to talk about Parkinson's disease in a Petri dish and tell you what we do in terms of stem cell research. It's not what you hear usually in the media, the stem cell transplantation. It's a little different. So the stem cells that we are using are called patient-specific induced pluripotent stem cells. Those are not the embryonic stem cells that we get from human embryos. So the whole ethical problems around that topic is not applicable here. So we have in the middle here the patient with the injury or disease. So we are interested in Parkinson's disease at the Institute. So we are collecting skin cells from patients from our clinic. So we take a biopsy, and we have derived 60, actually today it's 61 biopsies from patients that come through our clinic. We use those skin biopsies to expand in a culture dish. And then we bank those, so we have huge liquid nitrogen tanks where we can store those cells. And then we use a trick that's called epigenetic reprogramming. So in that case, what we do is we introduce certain proteins into those patient skin cells, and what happens then is they turn into stem cells. So we can basically rejuvenate the skin cells and turn them into cells that then can make any tissue type of the human body. So we can differentiate them into neurons, heart muscle cells, beta islet cells, and also into different blood cells. So you can basically make any cell type of the human body, which is very exciting. So this epigenetic reprogramming was developed about 5 years ago by a group in Japan, Dr. Yamanaka's group, and has been now used by a lot of labs around the globe, not just for Parkinson's disease, also for other common human diseases. So once we have those pluripotent stem cells in a dish, since we're interested in Parkinson's, we differentiate those pluripotent stem cells into neurons. And not just any type of neurons, but into the dopamine-producing neurons. The dopamine neurons are dying in the brain of Parkinson's patients, so that's the cell type of interest for our studies. And then there are two ways. So either you can use those cells then for cell replacement, which is not the focus here at the Parkinson's Institute. There are other labs that are trying cell transplantation. We're using those cells for in vitro research. So that means we are studying those cells in the culture dish, in vitro. We're not putting them back into a human brain. And then there are a lot of things you can do with it. You can study disease mechanisms, you can use those cells for drug screening, and also understanding human genetics and developmental biology. So this is more specifically how we model Parkinson's in a dish. So again, you take a skin biopsy from a patient, you isolate those cells, they are called fibroblasts, and this is how they look in a dish. So this dark piece is a piece of skin, and then there are two types of cells growing out. First, these cells are called keratinocytes, and these cells, these are long, skinny, tiny cells. These are the fibroblasts that we are then using for the reprogramming or rejuvenation of the cells. So after reprogramming, the cells look like this, and I'm sorry, it's a little dark. So what you see here is a colony, an induced pluripotent stem cell colony. And this aggregate of cells includes several thousand cells. So it's not just one cell. It's maybe 5,000 to 10,000 cells. So once we have those iPS colonies, we directly differentiate those into neurons. And there are several phases. So we start with the phase that's called neural rosette stage. So it's an early progenitor cells of the neurons. And then we differentiate them further into mature neurons. And the whole idea at the end is to use those cells, again, to study disease mechanisms, and also to use those cells and find new drugs that could help patients with Parkinson's disease. So when you start with a stem cell colony, what you do in the first step, you try to lift those up with an enzyme called collagenase, and then you grow those stem cell colonies as floating aggregates. So these are little aggregates, and we actually have them on a little shaker in the incubator. And then they shake for about 10 days. And during that time, we induce the differentiation into those neurons. After 10 days, we plate them down again on a dish, so they attach again on a plastic dish. And then they form these cauliflower-like structures, or rose-like structures. Rose is a little nicer, so sometimes they look like cauliflowers. And here they're called neural rosettes. And then manually we go in with a scalpel, cut those out, dissociate them into single cells, again with an enzyme, we break them up. And then they grow as single cells, and here we call them neural stem cells. So there are specific markers that have to be expressed to be able to characterize them as neural stem cells. That's why they are green and red here. And then you see here, it takes another month or even longer to then differentiate those neural stem cells into mature neurons. So this can take up to 4 or 5 months, if you will. It's good. So we chose a protocol in which we can take a break. Let's put it like that. So at this stage, we can also freeze and thaw the cells. And then from here to there, we have to continuously feed the cells. So there are no weekends. They also want to be fed on the weekends. So it's kind of, it's a very demanding process to make neurons. So this was done by Sally Mack. She developed this protocol here at the Institute. And what you see here in yellow, these are the dopamine-producing nerve cells. These are other very nice pictures from her. This is how the neurons look like. So you see the tiny dots here, and then long processes that form these very dense networks of neurons. They also express certain markers. So that's important to show reviewers that you actually have neurons that have certain characteristics. So that's why they're all green and yellow and blue. So they express GABA, alpha-synuclein, synapse. So those are all markers. So again, when you ask yourself, why are you doing all of that? And what's the use of this stem cell model? We believe that we can use these cells really for discovery of new disease mechanism, because there has not been any good model for Parkinson's disease. A lot of the cell culture models were from cancer cell lines. So this is really the first time that we have neurons in a dish that actually derive from a patient with the disease. So that's something very, very novel. Also, other models were really from rodents, from rats and mice. And rats and mice don't really get Parkinson's disease. So it's really important to have this new human cellular model to understand the disease. And here again, it shows kind of the how everything evolves. It takes a lot of time. The dopamine neurons here are standing at the end, but we're also thinking of making other neural subtypes that we think might be also affected in Parkinson's disease. For example, striatal neurons. There's also literature that the glia cells, the cells that support the neurons, might also have a role in Parkinson's disease. So we're also making those in addition. So you can do a lot of things with this model. You can also try to modify those lines. For example, some of the patients we have we have skin cells from have certain Parkinson's mutations. So you might have heard about LRRK2. So there's one common point mutation that can cause Parkinson's disease. So we have a grant to correct in these cells these specific mutations. So we go in with certain molecular scissors and can correct the problem. And then we can also study if this one mutation that we've corrected is actually responsible for the problem and for the phenotype. So that's very important. So you can do that for LRRK2, you can do that for other genes as well. And then here we believe once we have a valid phenotype and understand the disease mechanisms better, we can use those assays also for high throughput drug discovery. So that's the whole circle, how we believe this model is useful to understand Parkinson's and to help you guys. Okay, so this is a thank you note for all patients and volunteers that have put skin in the game. So at this point, we have 61 biopsies, so thank you all for participating in research at the Parkinson's Institute. We need you. Really appreciate participation of patients here. And it's, as I said, it's a very demanding program, so we have a lot of people helping and working on the weekends and day and night to make this happen. We have very, very good collaborators at Stanford University, Max Planck Institute in Germany, and also we have some industry collaborations, so here at Sangamo Biosciences. And we, of course, thank foundations and funding agencies that support this program. Thank you so much.